And welcome again to Cancer Center Grand Rounds. I hope everyone had a great 3-day weekend and are rejuvenated. I guess the unofficial start of summer has arrived. And we have, Renee, we have Grand Rounds, though, continuing through, I don't know if she's here, through the end of June, right? And then we take a respite. I'm learning Yale, so forgive me. And we take a respite in July and August, is that right? So the home stretch of the academic year. And I can't think of a better person to be here for the home stretch than Dr. Marcus Bosenberg. And as you know, Marcus is one of our leading physician scientists in pathology, dermatology, and the Cancer Center. Marcus is a co-leader of our genomics, genetics, and epigenetics program for the Cancer Center, and is a co-leader for the Yale Spore and Skin Cancer, which is moving forward as really a leading center of translational research for our Cancer Center. Marcus, as you know, has done sentinel work in leveraging mouse models as part of his postdoctoral training to informing cancer biology, and I think now informing novel therapeutics in melanoma, most prominently immunotherapy, and we're really pleased to have him today. His talk, as you see, is entitled Research in Anti-Cancer Immunology, How to Utilize Mouse Models and Human Tissue in Cancer Research. Marcus, thank you.
Thanks a lot. I think the goal for this Grand Rounds is a little bit different than most other Grand Rounds, where there will be a little bit of science that I'll talk about in the beginning portion of the session. I think I've got a whole hour. I think an out-of-town speaker had canceled, and I had agreed to step in. And one of the things I see, so I'm as a physician scientist with a background in pathology and in mouse models, is these are two of the areas that we get questions on about as frequently as any others, where it's not areas that everyone feels either comfortable or trained in. So the last half of the talk will be a little bit, first half will be sort of current mouse models, what's the state of the art for evaluating anti-cancer immune responses, and what can one expect or use. And then the second half will be in regards to how to utilize human tissue in cancer research, and this is sort of an overview of those two parts. And the reason for bringing this up is there's currently an efforts underway through the Yale Cancer Center and Yale more broadly to make it easier to identify sources of tissue and to optimize your own approaches to doing tissue-based research. Most of what I see, though, in person's request reflects sort of a not complete understanding of how tissue is optimally used in experiments. And what I'll show you is some approaches to basically generate preliminary data without having to go out away from your computer. You can do all of this stuff essentially from, in literally hours if you know what you're doing with it, and generate preliminary data for grants, generate figures for papers, give relevance to the pathways and genes that you're interested in studying for your particular area of cancer biology. So, but getting back to the first part of the talk, we've developed a number of models over the years for different purposes, mostly related to cancer genetics, and these are some genetically engineered mouse models. I'll also talk about some older models and how all of this is, including an area we haven't worked that much in right now, but it's a lot of confusion in this area as to humanized mouse models and how and what might be coming on those horizons. So you'll have kind of an overview more broadly of this. And I think I know of probably 15 or 20 labs at Yale alone that are venturing into the area of cancer immunology, and pretty much by necessity are using mouse models. We've supplied a number of them, but there's also folks doing this on a variety of fronts, also generating models certainly outside of us. And the translational approaches I'll talk about, we'll get to it in more depth later, but I really am trying to emphasize these in silico approaches, which we typically use ourselves rather than going to track down tissue and other collaborators that would typically takes months to actually pull off. This can be done literally in an afternoon. And then, what things do you really actually need to go and find tissue or fresh tissue for, and what goes into that. So the genetically engineered mouse models, and I should like to also give credit and acknowledgement to others at Yale, including Katie Politi and Nick Joshi, who both have strong interests in developing genetically engineered mouse models that will reflect anti-cancer immune responses, and are working very actively with some very elegant approaches in that. Now, some advances in genetically engineered mouse models is basically restricting the genetic changes just to the cancer cells that will form, rather than having a germline knockout, for instance, in P53 and seeing what cancers happen to show up, because it's typically not in the organ site that one would want to have. And there's many elegant models, both for Katie and Nick's work in lung cancer, but then also in other places, including pancreas and so forth. You can have models where you have a hit that happens sort of randomly in a subset of cells, and then you have normal versus cells that have genetic changes, which helps as a pathologist and as a biologist to determine what the effect of those changes are. Sometimes they're on in every cell, and it's a little bit harder, but not impossible to tell those changes as well. So some of the things that are nice about this is it's very physiological context, but the problems are that the latency is long, so you might have 6 months or longer for tumors to form. Sometimes only half the mice or 70% of the mice get tumors, so that complicates your analysis. And for immunology experiments, one thing we've found now is that by putting so many changes, even with the longer latency in mice, there's very few genetic changes outside of the ones that you put into the mouse. So if you put in a KRAS allele or something else along those lines, you don't tend to get more than 10 other mutations unless there's some kind of a mutator phenotype. And that's a bit of a problem for trying to evaluate immune reactions against cancer, which, while there's not an absolute correlation with the number of neoepitopes or new mutations, there is a correlation that's reasonably strong. And if there's very few mutations, it's typically a lot harder for the host or the body to mount an effective immune response. One thing I think that is very nice is that typically when you do grafted tumors with cell lines or other things that have been grown in culture, their mitotic rate and growth rate is almost unparalleled in human tumors. There's like Burkitt's lymphoma that might grow as fast as a grafted tumor, but almost everything else grows much more slowly, and that is part of the physiology of tumor growth. And so it's hard to get, if you can actually cure a genetically engineered mouse of a tumor, you're doing really, really well. So it's a high bar. So if you want to have an immunologically resistant, so-called cold tumor, this is sort of what you're looking at as probably the highest bar for that. And so you can improve or alter these models. Again, I think Nick and Katie in particular are doing some of these things where Nick has a very elegant system of having model antigens expressed at certain times, like versions of albumin or something else, where there's really well-characterized tools in particular genetic backgrounds to evaluate those immune responses. You can use a mutator allele. For those of you who are not aware, that colon cancer across the board is not particularly sensitive to immune checkpoint inhibition. However, mismatch repair deficient colon cancer is very responsive to let's say, PD-1 blockade. So you can put in, for instance, like a mismatch repair deficient allele or a poly allele or something along those lines. And we'll talk a little bit more about chemical carcinogenesis, which still remains a useful approach. And so, speaking of that rather quickly, Bob Schreiber out at WashU, among others, has used a chemical carcinogenesis model where they inject the MCA, a polycyclic aromatic hydrocarbon, into the flank of a mouse, and 100% pretty much of mice get tumors. The latency can be fairly long, and if you induce the tumors in rag mice, they tend to grow and keep on growing. However, if you induce them in wild-type mice, occasionally you'll actually get regressions of those tumors that initially formed. And so what they've done, instead of just keep on injecting more mice with more carcinogens, is that they actually develop cell lines from these and then inject the cell lines into wild-type hosts. Sometimes they've derived them from immunodeficient mice like rag mice, and then go back into a wild-type mouse and show that it's rejected. They've also done some work characterizing these particular lines, and there's a high number of somatic mutations in those lines, and they've done some very elegant work in actually finding the neoepitopes that are present in, it was a lot of work to do, and showed that by immunizing with peptides to those particular neoepitopes, they get an enhanced response in mice that hadn't been previously exposed to the tumor. So some very elegant sort of field-defining work out of that the Schreiber lab using these chemical carcinogenesis-derived lines. So while the industry folks, let's say Merck, Bristol Myers Squibb, and others, what do they actually do when they're looking at developing their own preclinical data for immune-based therapies? And they typically use there aren't that many mouse lines that are possible to graft into immune-competent hosts. Most or many of the few that are there will grow in a background called C57 Black 6. Others might be BALB-C and so forth, but it has to be matched to the strain of interest. And a couple that are widely used are the B16 melanoma cell line, and then these two are colon cancer lines, and actually probably more reproducibly exhibit stronger responses in experiments evaluating anti-cancer immunity. Again, ironically, being colon cancer not having generally great responses, but these apparently have enough epitope burden that they allow for reactions to occur. So why do people use these things? And this was also true even in the era of targeted therapy, where genetically engineered mouse models weren't really the favorite of pharma because it's expensive to maintain and to use those mice for broad screening and large experiments, unless they clearly work better than other things, which sometimes they do. So these syngeneic models are easy, cheap, and fast to use. You can generate data reasonably quickly. Typically, the injection, even for these colon cancer lines, are done subcutaneously. That does certainly have its limitations. Tumors tend to grow fairly quickly. For B16, it's typically 2 to 3 weeks, even with 10,000 cells injected, a relatively low number. They grow out very, very fast, which actually does have implications for how to interpret your anti-tumor immune responses. It may be too fast for an effective response to be generated. And then again, they're variably immunogenic, and typically, for instance, B16 has pretty poor responses to immune therapy, so you could also look at that as another opportunity for making cures happen. And one of the things that's typically happened, as a former editor for a melanoma journal, typically one of our requirements was if you had a cell line based result, that we would require that you have at least two, not just one example of one of these. And most of the tumor immunology literature historically has basically had one model, like B16 or MC38, that was the workhorse for a particular paper. And it's really hard to say, is B16 like MC38? These two are reasonably similar, but I think the host strains for both of these are different, so it makes it a little bit complicated. So generalizing from these models and trying to interpret the preclinical data in terms of predicting responses in clinical trials in humans is not always trivial, and is an area that should probably be optimized a bit more. So another problem with some of those lines, for instance, all three of those, is that the driver genes, even with exome capture and sequencing, aren't always really that clear. I mean, the example I would use, lung cancer, melanoma, two areas that are typically looked at for anti-cancer immune responses, there's always a subset of those tumor types where it's not really clear. There's no BRAF mutation, there's no RAS mutation, what actually is driving that particular cancer, and it may be a combination of things. In the case of B16, it was shown that when you take away endogenous retroviruses, that they actually may be contributing to the pro-tumor growth phenotype, at least in part. And then they start out with for instance, like MC38, you have a relatively high mutation burden. So if you wanted to have a low mutation burden comparison, like a parental line that actually grows out and doesn't have a response, it just doesn't exist along those lines. So you start out with that's why the Schreiber paper with Tom Schumacher was really quite elegant. It's the only one I know of, I think, in 3 or 4 years down the line, actually identifying the antigens that have resulted in an immune response is not trivial to do, and we still don't know how many there are. So you can find 1 or 2, and you can find with tetramer-based assays that there are T cells in that mouse that do respond to that particular peptide antigen in the context of a particular MHC presentation, but you don't know if that's the only one or if there are more, and there's other approaches that will probably be needed to address that. So, ways that these models could be improved, and things that you can also do even to some extent on your own, is that, I'll refer, this is our work, at least coming up in a little bit here, is that you could make models based on the human relevant genetic changes. There's also interest, for instance, in melanoma to use targeted inhibitors like BRAF inhibitor or MEK inhibitor in conjunction with PD-1 blockade. And if you don't have a BRAF mutation in your melanoma, you can't really do that approach because it won't work. And it'd be nice to have more than one, sort of this idea of not just having a one-off experiment. And ideally, you'd be able to look at antigen-specific responses to trace initial presentation and then exhaustion or non-exhaustion of different cells along the way. Imaging techniques that allow you to see tumors in a deeper location are pretty much not required, but you can look at, if you don't know that the tumor is growing initially, how do you know that it's actually responded to a particular intervention? You really have to be able to say that, A, it was this size or so initially, and then it got smaller as a result of something that you did. And it'd be also nice, because in humans, one should never forget the good work that surgeons and radiation therapists do in getting rid of individual tumors. So the cause of mortality due to cancer is, overwhelmingly due to metastatic disease, yet almost all of the modeling we do is subcutaneous, and which is usually a typically easy surgical site to access. So is it different if the tumor is present in brain or in lung or in other locations, in lymph node and so forth? And most of that we don't really know. And so some approaches that people may be aware of, as an analogy, work in humans. You can do this in mice too. Bill Hahn had made a series of lines where it was looked at to see what the minimum number of effects to make something immortalized and transformed. You could, with CRISPR now, it's so easy to edit primary lines that presumably you could make a series of different mouse tumor types, either from mouse embryonic fibroblasts or from other cells. For instance, like melanocytes are not particularly hard to grow, and then sequentially CRISPR in different hits to see closely related lines. I don't think that's really been done particularly comprehensively, although I expect it will be over the next few years. It's like, oops, sorry, my fault. And so, I'll talk about humanized mouse models a little bit here, and one of the things you should realize is I would argue that perhaps the most advanced version of humanized mouse models that have been developed anywhere are, through the Flavell lab and the Mr. G mice, and some of you I'm sure have heard talks about those mice, where they've knocked in a number of human cytokines into the mouse loci so that the signaling between immune cells was a little bit more physiologically human-like. And one of the things that those mice tend to do is they tend to retain the myeloid-derived cells in the tumor microenvironment much better than the more typically used NOD/SCID IL-2 receptor gamma deficient mice. These mice have no B cells, no T cells, and no NK cells, and are kind of sort of the standard PDX host for a number of laboratories. It should be noted that nude mice also work reasonably well for these experiments. If the tumor grows, then there wasn't a problem with B cells or NK cells, from the mouse side. But typically, these NSG mice are kind of viewed as the standard. There are other approaches around this, and there's some intellectual property issues about using Mr. G mice outside of the Flavell lab. I will leave it at that, and or contact Dr. Flavell if you are interested in doing experiments with them. But I think there are some other approaches that may be of interest to know about. And typically, the goals have been pretty high with this, where you have an immunodeficient mouse, you have tumor either as a chunk directly out of a patient that goes into a mouse, or you have homogenized cells that have never been cultured out of a patient that goes into a mouse, or you have short-term cultures, which are just pretty much tumor cells, but that were as individual cells. And then typically what has been done is that there's been efforts to reconstitute the human immune system back in that mouse. Ideally, you might have mobilized hematopoietic stem cell precursors, perhaps by GM-CSF in a particular patient, and you match that patient's own mobilized hematopoietic stem cells with their tumor so that the HLA context, everything else is exactly the same. You pretty much have to have a clinical trial. There's a huge amount of work to make something like that actually happen. There are people who are doing that, but it's not trivial to do. So then another thing people will do is to take a single source of cord blood that may or may not be fully HLA matched, and then they'll use that as the source for hematopoietic stem cells. So it's like an allograft or something like that, where you're going for a different source for the hematopoietic system from the tumor. You can take patients' peripheral blood, and this has the potential advantage that there are already educated T cells that have been had antigen presented from the tumor that then they can react to. But they're typically present in low numbers in peripheral blood. And then there's the entire repertoire of T cell receptors that are in the peripheral blood that could react with other things. So, and I'll tell you in a second why that's important. And a last approach that I'll mention, which I think has really been used successfully also here at Yale, Sue Keck and Ryan Sal in her lab have done this very nicely, and others I think have as well. It's sort of like a Steve Rosenberg approach, where you take both tumor and you take a portion and you culture out the tumor-infiltrating lymphocytes and you expand them markedly. You put the tumor back in mice, and then after the tumor is established, which is larger than most tumors you can reject, you can put somewhere between 100,000 and 10 million TILs, tumor-infiltrating lymphocytes, back in. And Sue and Ryan have seen full regression of almost 1 centimeter tumors using this approach without the graft versus host disease that one typically sees in approaches where you're reconstituting the human system in mouse. What typically happens with more generalized approaches is the mice doesn't live more than 6 weeks because the human immune system eventually ends up phagocytizing a lot of the red blood cells, and you have a mouse that's anemic and expires from that or from other causes, which aren't always particularly clear, but your limitation time-wise certainly exists there. So I think that's a very elegant approach, which will be very, very powerful moving forward. There are people who are trying to come up with approaches to this where there's some kind of human thymus equivalent, so you can actually have the sort of induction of tolerance and other things that are there, but I haven't really been knocked out by the success of those approaches to date, although there is a lot of interest in this area. Something to keep in mind too is people always talk about or not always, but frequently say PDXs and humanized mice. This is the future. It'll be the future when it actually shows something unique and new, and that's also true for the other models. So I think you have to look at what purpose you're using these models for. Is it for hypothesis generation? Is it for mechanistic understanding? Is it for evaluation of a potential therapeutic that you want to decide whether or not you're going to go forward and spend a lot of money on clinical trials for? I think those are all important endpoints to distinguish, and it hasn't really been done in those ways. So I've mentioned that with the humanized mice that it is quite hard to do this. There's a whole apparatus that kind of has to be in place. The graft versus host disease is pretty typically limiting. I mentioned about the thymus, and that after the first generation, typically myeloid-derived factors don't reconstitute in the tumor microenvironment. Or there might be certain subsets of T cells that don't tend to come back after the first round, or even after a little bit of growth. Like the relationship or number of Tregs seems to be less in some of these models when you do the TIL approach. So that may actually have effects which aren't that clear up front, because maybe some of the myeloid cells are required to retain them. So it's a complicated thing, but I think still will be very, very interesting. And there may be human-specific immune mechanisms that are at stake. Perhaps these could be avatars for patients, that you actually do this approach and you see that if you use a STING agonist with PD-1, all of a sudden it works, and that may be true. But until we see that, I'm not sure that we'll know that. And that's the hope, is that it's predictive at some point in time. I kind of am excited about it for the mechanistic possibilities. If you have tumor cells and TILs both separate, you could presumably do CRISPR on either side of those things, and you could actually genetically evaluate some of the things that are responsible for human immune responses, which are very, very difficult to do in other ways. So this is a little bit more into our stuff. I think it's a general useful thing as to look at how people typically evaluate preclinical responses. And I think people will say, "Oh, none of the mouse like Ira Melman, who I have a tremendous amount of respect for." would joke about, like, why do we even need the mouse models? Well, let's just go with our clinical trial and see what we see, because they haven't been predictive. I think part of the problem with that is what people tend to interpret the data as and what they're happy with in terms of data. So typically what's done is you have a tumor volume plot. So you have tumor volume is the y-axis, time is the x-axis, and you take at various time points what the volume is for different tumors. And if there's a separation of the curves, there's a significant difference between the two curves, and we'll look at that in a second, that's viewed as success. And I have some issues with that as a general approach for making decisions. Overall survival is usually based on, in like a Kaplan-Meier survival plot, when tumors from Iacoc's regulations reach a volume of 1 centimeter cubed, you have to euthanize the mouse. So that's the overwhelming reason. You can't typically just let a mouse die of tumor. That's viewed as unethical. So when it reaches 1 centimeter in volume, that's when the experiment's over. The curve that's doing better, that has slower death, typically always dies of tumor as well. It's just a couple weeks later. So it's not always, especially in the B16 experiments, it really can be 4 days or so. That's a separation of the curves, even though it's significant. And there's a lot of correlative work that involves flow cytometry, but typically these are performed at the end of the experiment. I, as a pathologist, get consulted after the experiment's over. People have these large ulcerated tumors that they've done flow on and so forth, and it's really not the best way to do the analysis for a couple of reasons. And I'll hopefully illustrate that here. So here you have 3 different tumor volume plots. Let's say this is the control, how the tumor normally grows. Let's say we gave PD-1 blockade or CTLA-4 blockade over here, and this is the curve where it's typically slower throughout the growth period. It never really gets the full sort of exponential growth phase. And then there's another plot over here in red where it looks like it's relatively stable for a while, and then it grows a lot faster. And if you actually take the tumors here, which is typically done, I would argue that this red curve, taking a tumor here versus taking over a tumor here, you'll see 2 completely different answers. And so I think you have to just be aware that, if one talks about acquired resistance, this is perhaps a curve that you might predict where something actually was somewhat controlled by a particular intervention, and then it changed and grew out rapidly after that. And once that event's happened, it's presumable that it looks like the other 2 curves in terms of how fast it's growing and other things in the particular tumor. But up to that point, there might be something happening here you just don't realize. So you have to build in additional time points to look at things when the biology is actually happening and be cognizant of it. And so, as I was saying before, this is not unusual to be viewed as a success in preclinical testing. In clinical trials, this is progressive disease, and this would not be a success. If by RECIST criteria, that doesn't really, that's a failure. And so I think those are just basic disconnects in how we interpret preclinical data that should probably be addressed at some point in time. And so what I would view as sort of like your dream preclinical response is that you actually grow a fairly large tumor, I was talking about with those humanized mice with the TIL therapy, almost up to as large as you can get, half a centimeter, maybe close to a centimeter. You initiate your therapy, giving TILs or PD-1 blockade. The tumor may not regress immediately. This happens in human patients where tumors have sometimes unusual responses where they'll grow for a while and 6 weeks later they'll shrink. But then at some point in time you see a response where the tumor is getting smaller, and then the tumor goes away, and for the lifetime of the mouse, whatever you're going to decide you want to follow that mouse for, the tumor does not recur. And if you were to challenge the mouse with a larger burden of, let's say, syngeneic cells or re-induce another gem tumor, that would even be more efficiently rejected. That's sort of the characteristics of a memory response that we're very familiar with antiviral immunity in other areas along the way. So that, I kind of view this as a gold standard. It may not always be possible, but I think that's what one has to be shooting for. So with that in mind, I know a number of you are using some of these lines, and others may perhaps, and these are available for anyone at Yale with really pretty much no restrictions. We've made a number of syngeneic lines that are mouse melanoma models on a C57 black background. We, being Katie Meath, the former graduate student, did 99% of the work associated with it, or 90% of the work associated with this. And we tried to make a number of genotypes. We're, I guess, through 6 here. Some of these are still happening. With the idea, and these are all published sort of studies in terms of what the phenotype of these tumors were, and that was the reason for doing it initially. But the thought is that genotype might affect your immune reaction response. And There have been a couple papers, actually quite prominent papers, which some of you are probably aware of, using beta-catenin being an indicator signaling, Wnt signaling of perhaps an immune-poor environment, which was basically directly based on this model here, which we had developed a few years back. And then there's also a paper out of MD Anderson, which I'm also on, looking at PTEN deficiency and its role in affecting immune responses. And I'd say that I don't really know how to interpret both of those papers entirely. I'm not saying the data is incorrect, but they tended to look at relatively small changes in terms of phenotype, not a complete response versus a not complete response. So I think that those things can be modifiers, but they're not the dominant phenotype, and I'll tell you why I think that in a second. So these are the parental lines. I think all of these form tumors in C57 black mice without problems, essentially. There's a couple that are a little bit slower. I'd say what we know now is they probably have between 200 and maybe 500 some SNVs that are coding. So they have, by exome capture data, we've only looked at a couple of these so far, there are actually a number of mutations that could potentially be immunogenic. But they allow for infiltration of immune cells, but the tumors tend to grow out in mice. So as a control, they're more immunogenic, I think, than most of the genetically engineered tumors, but not as immunogenic as they possibly could be for an efficient response. So this is the work of a medical student who's still a medical, or I don't know when graduation is, but Jake Wang, who is in the lab and will be around as a dermatology resident in the future at Yale, who took the approach, along with some other lab members, of UV mutagenizing some of these lines, and with the idea that we would introduce more mutations in particular lines. And the second part that was important for that was to actually perform a selection of single-cell derived clones. So I'll explain sort of a control experiment for why that's relevant. Most human tumors are thought to be, and based on the genetics, are single-cell derived tumors, where you have one or two or more genetic changes present in pretty much every cancer cell that's part of that cancer. It isn't always that way, but generally that is the process. And so what Jake did after he derived, this was YUM is Yale University mouse melanoma, and then the ER is exposed to radiation. I didn't come up with the YUMmer name, but it seems to catch. And so in C57 black 6 host, the parental line grows out, but the mutagen line grows for a little while and then is spontaneously rejected when 100,000 cells are injected as a starting injection volume. In immunodeficient rag mice, both tumors grow out at similar rates. And I think this illustrates a couple of principles that I think had been noted before, aren't generally appreciated always now. So in addition to the RAG, so the reason for doing the RAG experiment is it's typically a control experiment to whether or not the immune background makes a difference for whether the tumor grows or not. So this is like the Bob Schreiber MCA experiments. These would have been typical controls done there. Another way to do that is to do depletion of CD4 and CD8 cells using antibodies, and those are down here, where we see a bump up from the control. Doesn't bring us all the way back up, but when we have both of them together, we see pretty much a wild-type tumor growth, or a YUM 1.7 tumor growth. And depletions aren't always perfect, but this also helps establish that this is dependent on T cells, not just potentially B cells. With the RAG have both deficiency in B and T cells. So one of the things that Jake had noticed was that if we injected less than 100,000 cells, the tumors tend to get rejected pretty much nearly all the time. But when you went over that starting number, that we actually had outgrowth for the most part. So 500,000 cells, we typically had essentially all the tumors grow out, and depending on where the cells were in their population doubling sort of status, at lower numbers of population doublings, they definitely, sorry, jumping ahead here, were rejected spontaneously. Now that gave us a situation where we thought that the 500,000 cell injections were having an exuberant immune response as a pathologist looking at them, but that they might be more amenable to adding a therapy because they're right on the brink of being potentially rejected. And so, and here's a close-up of the lower cell number injections and what kind of tumor formation we saw. So Jake and others in the lab have then used CTLA-4 blockade or PD-1 blockade in the context of the 500,000 cell injections in that YUMmer model, And if we don't give treatment, the mice die in a little over 30 days for having tumors that are 1 centimeter in size. And that when we do the combined treatment of both, we have cure 100% of the mice. CTLA-4 pretty much almost cures all the mice, and PD-1 blockade cures about half of the mice, a little bit less sometimes, a little bit more sometimes. And while when we treat the parental line that isn't mutagenized but has 300 SNVs on its own, we don't really see much of an effect, certainly at least with the PD. I think as the combined, it's off the slide, but PD-1 and CTLA-4 blockade. So some of you might be wondering, I'll preempt your question, why is CTLA-4 better here than PD-1? I think part of the issue is that the CTLA-4 antibody that's used in a lot of the mouse studies tends to actually induce antibody-dependent cellular cytotoxicity, is the thought, and CTLA-4 is highly expressed on Tregs, so this is probably somewhat a Treg depletion therapy, and we know that when we deplete Tregs by different methods, genetic methods involving diphtheria toxin receptor and administration of DT into mice that have, FOXP3, Treg-driven diphtheria toxin receptor, we can actually reject pretty sizable tumors just by that therapy. So Tregs are very important in this response. So I've kind of talked about two probably related, but not necessarily absolutely related, or three actually different responses, where you have spontaneous rejection at 100,000 cells, you have outgrowth at 500,000 cells with this particular model, and then we have responses to checkpoint inhibition that actually I'm pretty happy with. And once we have a mouse that's been cured this way, we can actually give, without even other therapy, 20 million cells, not just 100,000, and they'll reject it fully with that number in mind. So this is one cell line. I was telling you not to believe anything from just one cell line. So there's additional models that are in place or that we have developed that will be things that one can do to validate discovery, for instance, in something like YEMR 1.7. We also found that after we made the line that when we were looking at allele frequency in the exome capture that everything seemed to be 0.25, and we thought, okay, either it's two clones, because you should have half if you have a mutant allele, and so it's a tetraploid line, which is not optimal for certain things, but maybe it allowed the line to survive UV mutagenesis better than a diploid line because the allele frequency was lower. We don't really know, but we have diploid versions now that we're working on. So I thought it might be of some interest to actually talk about what do we see, and I think I would emphasize too, if you're doing these types of experiments, that even timed euthanasias every day, for the course of 30 days isn't that many mice, a couple tumors per mouse. It really gives you a much better picture than just looking at the end of the experiment. You wouldn't really know what just happened. I won't talk about it much here, but B16 as an example, there are practically zero tumor-infiltrating T cells in the actual tumor. Around the rim, around the outside of the tumor, there's about 30-fold or 40-fold more, at least, just outside the tumor, but not in the tumor. So it'd be nice to know along the course of tumor growth what happens. So what do we see, and this is kind of a summary, is that by 4 hours we see macrophages in the area of the injected tumor cells. We've labeled the tumor cells with GFP, which is antigenic to some extent, but in the YEM 1.7, those tumors are not rejected with just GFP, so you can use it to at least mark your cells. Then at about a day, we see neutrophils, and we don't see any lymphocytes before about day 7. And this is in concert with what one might see with viral immunity as well, if you're looking at a certain site and so forth. And we see B and T cells arriving roughly at the same time. And as a pathologist, there's killing that I can see by roughly the next day. It's not like the whole thing is just being necrotic. When I give CTLA-4 therapy, or look at mice that have been treated with CTLA-4 therapy, it almost looks like an infarct. There's actually geographic necrosis where a whole area is dead, and it's dead earlier. So I don't know that the killing mechanisms are identical, because the histology certainly is somewhat different between the two. We need to have a little more density in those time points. But in the escaping tumors, the YEM 1.7, it's hard for me at day 9 or 10 to tell the tumors that are going to get rejected from the ones that are not going to get rejected. And I think that's an important part, because obviously we'd like to be able, I'd like to be able to do it visually, but what's the science behind those two differences? And I think we're certainly looking at that, and hopefully other people will have some more ideas and thoughts about that in their own work. And so one of the things that I'd also emphasize, and this is also sort of plugging the tumor immunology laboratory that Kurt Schalper is running. We do a fair amount of histology ourselves. but that if you did flow cytometry, these tumors up to here are all very small. You wouldn't have adequate number of events to actually have, these are about 3 millimeter tumors or something on that size. So you wouldn't be able to tell particularly accurately, and you wouldn't tell whether the tumor cells are inside or outside. So I think doing these, and for those of you who don't know of Kurt's work or other work, and David Rim obviously as well, where you actually can do multiple markers and quantitate the relative expression of different things in particular individual cells and actually get almost flow cytometry-like data out of it. Those approaches are pretty labor-intensive as well, and also fraught with a lot of controls and so forth. So I'll show you just some standard immunohistochemistry that's done by the Research Pathology Laboratory, which is an outstanding facility to do your staining here at Yale. This is a tumor at day 5 in the 500,000 cell injection tumor, Yummer 1.7, with the 100,000 pretty much almost all the cells you're looking at here are tumor cells. And this is the edge of the tumor, and there's a few myeloid cells kicking around the edge. And I'll also point out that there's very, very few mitotic figures. You may take my word for it here, but up to day 10 or 12, the mitotic rate is about 1/10 of what it'll be at day 20. I didn't realize that in different tumor types that it would range that much of a difference. This is the 100,000 cell injection at day 15. And for those with pathology training out there, you might recognize these nuclei as being quite a bit bigger than their neighbors and would be putative tumor cells. This is where the GFP is kind of handy, so you can actually see this is what was a larger mass before. This is what's left. We typically see the cells being isolated and then presumably killed as a result. The GFP staining was pretty exuberant in this case. This is probably GFP being taken up by adjacent phagocytic cells, but nonetheless, you can see cells in various stages of dying in this particular image, and that's probably near the height, the tail end of the height of what's going on. So if you actually inject more cells, and this is the tumors it's growing out, I can show you right here that there's multiple mitotic figures already. You can see in this image, there's vat angiogenesis, and we want to figure out if that link is very closely coupled or not. And one of the things, it seems like this is like the first image I showed, which was pretty much a wall of tumor cells. And it turns out that this is a macrophage or histiocyte stain for mice is F480. And it seems like every single tumor cell, these big round guys here, is surrounded by a myeloid cell that's either providing a surface interaction, who knows, whatever else, but almost like a network of cells that may be supportive or immunosuppressive locally. And what's striking about this, and work that hasn't been published, but was done with Jeremy Jaycox in Ruslan Metsitov lab. Jeremy will be a medical intern and resident here, I think, next year. He's an MD PhD in the program here. That if you do 2-photon imaging in the core that Ann Haberman runs here, these cells are moving all the time. In a half an hour movie, you see all sorts of movement, and they're just going also, it's like you freeze them in time and they're always around a different cell, but they're sampling the environment extensively. And it's even more impressive when you give a CSF1 receptor inhibitor, something that's required for these cells to normally grow. But I think it's a dynamic microenvironment, and all the stuff I'm talking about for histology will probably need to be repeated with live imaging at some point in time in the context of the tumor when that can be done. Skin tumors are pretty easy to do that because you can get them at the surface, and you can image over hours in a tumor. And this is just for kicks, is an image of FOXP Tregs in that large established tumor. And in our hands, probably about 50 or 60% of the lymphocytes total in those large outgrowing tumors are FOXP3 positive. In the middle. Outside, it's less so, but in the middle, that's what we see. I'd mentioned before about the need to do work at metastatic sites. Katie had also developed approaches really taught by Don Wen's lab, who had done this work initially when Don was a postdoc with Joan Massaguet, but doing left ventricle injections, and then this is a luciferase or IVUS imaging, a bioluminescent imaging. Directly after the injection, you can see staining all over the mouse, which is kind of cool, that 100,000 cells, and you can see the light coming from them all over. After a couple weeks, you can see multiple tumor masses. This is typical in a mouse that hasn't been selected before. And then in a line that Katie had developed, she could eventually get brain-specific metastases by culturing out the cells that went to brain and then repeatedly injecting them in the left ventricle after they've been antigen or pathogen tested before going back in. And the pattern you get is actually like the multi-metastatic pattern you might see that happens in patients rather than sometimes the direct injection approach, which gives you one dominant mass as a result of that. I'm not going to talk a lot about this, but there is a need for these approaches moving down the line to look at the biology of immunology at distant metastatic sites. And so we may not actually get too much of it. We'll see if we get the tissue stuff. But anyways, how can these models be improved? I talked about variable mutation burden. I think we don't really know the role of epigenetics very well at all. It'd be very nice to do antigen-specific responses the way the Schreiber lab has done to some extent, but to really know what those antigens are, and we're trying to do some of this with CRISPR-based approaches. The others are doing this as well. The functional evaluation of the responses is probably what we're spending most of our time doing now. Like, are NK cells required for the response? Are B cells required for the responses? In these models, which may or may not be representative of the full picture in humans, but it's maybe true that one patient may not represent the response in another patient as well. So I think you need to know the full spectrum. We had a really nice single-cell cancer biology retreat on, I believe, Thursday. There's a lot of techniques that are coming out that allow us, for instance, to determine how many types of myeloid cells are there in the tumor microenvironment. People talk about M1, M2, MDSCs, but in an unbiased way, how many are there actually? And I think evaluating the new and next generation of combination therapies to give guidance to clinical trials will be relevant. So I'll let people take a quick breath, and those of you who don't want to do human cancer tissue-based research, you can. I will not be offended if you leave and have other things you need to do. But those of you who want a relatively quick and I think painless approach to developing preliminary data or evaluating hypotheses at 1 in the morning when you have a thought that you just need to have addressed, this may help you out. So why bother? I think the typical structure, or a not uncommon structure for NCI grants in the past and even now, has been you might have a basic science aim, you might have a mouse modeling aim to go in vivo, and then you have to have some kind of human relevance. And if you don't have that in your grant, you're going to have issues in terms of getting it funded. And for papers, that's also true. We publish stuff on the mouse-only side of things. We typically have to show stuff either in human cell lines or in human tissue. So I will give you some general tips and then a little bit of an overview of how to approach this at Yale. It takes more time than 15 minutes to actually go into all of the IRB and things like that if you really want to do your own banking and so forth. So one approach is to get your own IRB approval, talk to surgeons about getting cases from their patients, having consents done, develop those as collaborations so they're also intellectually involved, and then you have a pathologist that you either work with or a facility that can do immunohistochemical stains, someone to do the analysis and quantification of data. There's a lot of players in this picture, and it's quite complicated. I do a lot of this stuff a lot of times. Sometimes it cannot be avoided, but if you can avoid it and your questions are more basic and you have some flexibility as to what the human correlative data might be, here's a couple other approaches. So if your cancer type, which applies to nearly all of the cancer research that happens at Yale, has been analyzed by the Cancer Genome Atlas, then you're in luck and there are possible things for you. If it doesn't, you have to find other data sets which are not as robust as TCGA. And I'll talk about something else which is called the Human Protein Atlas, which I think is very handy. It's probably a little handier for me as a pathologist than if you haven't looked at tissue, but it's kind of, I think, fun and allows you to look at things really whenever you think of them. So those of you who don't know the Cancer Genome Atlas, this is a large NCI effort. I think I may have been the only Yale site that submitted tumors, and this was in melanoma. But it had an approach where you had to submit frozen tissue and blood or normal tissue from the same patient. You had to give clinical annotation of the cases that you had, and the histology quality assurance is better than anything I've ever seen in a paper. So there's frozen sections on the top and bottom of the chunk that you submitted, and they had a third-party pathologist, as well as your own site, review the histology to make sure that it was representative of tumor. Most papers, I can tell you, especially ones in prominent journals, do not do that. And so what they did typically was mostly exome capture and sequencing of tumor and germline. They did some whole genome as well. They did RNA-seq on those, sometimes microRNA-seq. They did the Illumina DNA methylation chip with the 500K chip. So they have methylation data. For those of you who don't know what reverse phase protein analysis is, think of like doing 200 Western blots on a small bit of tissue from a cancer. So this gives you protein phosphorylation data and actually protein data. It's reasonably robust. I'm not sure it's absolutely perfect every time, but it's there if you wanted to look at it for most of the tumor types. And again, the clinical annotation. So what would you do with all of this information? Well, how do you access it? So one place you can get it from is there's a place, I think Sloan Kettering had set up, I believe, CBioPortal, but those of you who haven't checked this out, it's a reasonably user-friendly website that has a number of tools, analytical tools that are on the website, and has not just TCGA data sets, it has other data sets as well. So as a place, and especially for grad students and postdocs, and you work in cancer biology, you probably should learn how to use some of these tools. COSMIC is probably the largest repository for cancer mutational data, so you can look at where particular mutations happen in your gene or what cancer types tend to have mutations in your gene. So as a melanoma researcher, every year there's new BRAF mutations that are in like Langerhans cell histiocytosis, rare tumors where they do analyses, and that'll show up in COSMIC. You can download after you apply for TCGA data, and you have to have bioinformatics support and maybe someone who can program an R or whatever to do all the analyses. This is the approach that I've been lucky enough to do, and I'll talk about some things in those lines, but that I think is the most powerful way, if you can do that. I'm not going to put Dr. Fuchs on the spot, but this is something maybe we could think about cancer center-wide. It's relatively straightforward to support, and the answers and questions can be turned over relatively rapidly. So if not, maybe there'll be a pipeline of people who would be willing to collaborate, if not directly. And then you don't have to worry about getting the download of the TCGA data and stuff. Here's an example of one of the things you can do. So you have a gene of interest. Let's say HIF1 alpha in melanoma. Does it play a role? So what you do is you get RNA-seq data of HIF1 alpha. It's 500 cases, and then you say, out of all those 500 cases, you have a level of expression from HIF1 alpha, and you have a overall survival data, which is usually pretty robust here. And you could do top versus bottom half, but you don't know if maybe 10% have really high HIF1 alpha, or is it 20% along the way. So the cutoff point is something that you can kind of look at a little bit. What's often done, which is not the best approach, is you take the top 25% and you compare it to the bottom 25%. Well, the problem, if you actually had a clinical test that worked like that, it means you're throwing out the middle 50%, and you won't even talk about them as an example. And so typically, if you have a cutoff, whether it's 10 versus 90 or 90 versus 10 or something in between, it's a little bit more actionable. But anyway, this approach is pretty easy to do. And if possible, you should use stage match data, like for melanoma would be stage 3, might be a relevant stage. And what this tells you in your introduction of your gene X is that based on the survival data, this is from a manuscript that Goran Misevic is an outstanding graduate student that I had who's still at Yale as an PhD student who developed a lot of the approaches that I'm talking about, and this is his data. But DNA methylase, methyltransferase 3B in stage 3 melanoma had a significant survival disadvantage when it was highly expressed versus a different methyltransferase. And then he also did, just to show you sort of the other examples, is that we found, and I'll talk this a little bit, not much, but that one of the microRNAs was probably methylated by this. And then the hypothesis would be that if you had high methylation, corresponding with high DNMT3B, that you might have worse prognosis, and we actually did find that. So it's not just RNA levels. You could do methylation sites. You could do anything else along those lines. You could do mutation burden as well, or a mutation type. But this is a typical first figure or figure in an R01 grant or in a paper when you have a gene that may not be as well known and you're trying to argue that it's actually useful and important in a particular cancer type. So another analysis is something which I'll call here as expression associations. And it's pretty straightforward. What you're taking is, let's say, HIF1 alpha, and you think HIF1 alpha is associated with hypoxia. So if you go in melanoma and you say, I'm going to compare HIF1 alpha to every other of the 30,000 genes that are there and have a correlation coefficient. Every tumor you'll have two values, HIF1 alpha and the other gene. And then I want to make Pearson's probably the way to go for most of these things, but you can do either sort of correlation coefficients. And then if you had a perfect correlation of the two, the one would be the row that you would get out of that. And if they were perfectly anti-correlated, it'd be -1. And so if you have a rank order list, and I'll show you a couple examples of this, you can actually take top and bottom and do gene set enrichment analysis, which might give you pathways that are relevant that your gene might affect, like HIF1 alpha. You'd expect it to be hypoxia would be a positive control for that. So this is a very powerful approach when you're kind of trying to find a mechanism in a particular cancer type of a gene. So here's an example with PGC1 alpha. It's a mitochondrial gene. These, you'll have to take my word for it, that's an enzyme in the TCGA cycle. And here's actually from a funded PO1 project grant, some hypothetical genes that would be correlated with PGC1 alpha, and some downstream effectors are there, some known upstream effectors are there, and other things that were thought to be there weren't really that strongly correlated. Here's the example of like gene ontology terms with PGC1 alpha expression in melanoma, and you can see that it really does correlate really strongly with mitochondrial gene output. Another example, which might be of interest to folks in the room, would be with TCGA data, how do you determine whether a tumor is inflamed or not? So you could do different versions of CD3 for T cells. This is the zeta chain of CD3, which is the CD247. And this is kind of stunning. If you look at the row values here, you'll never see correlations this good in pretty much anything you do biologically. There's 391 genes with a row of over 0.7. So what can you conclude from that? Well, in melanoma, you could probably pick any of 200 genes, and that would tell you what the T cell infiltration is, because that level of correlation is really, now it doesn't tell you about the state of those particular T cells, or even if they're 4 versus 8 or something along those lines. You probably could do some work with that, but I found that to be quite striking. And then the other thing you can do is to say, what's the strongest anti-correlated genes with T cell infiltration? And you might find things that exclude T cells on a regular basis as a result of that. And you can do that in any cancer type that's of relevance to your own. And one example of that with Chin Yan, who's a great researcher in our program, we're looking at KDM5B, and this is how we try to get some human translation related to a spore project that we're working on together. And also I gave the example already about mechanism. I'm not going to belabor that here. So in the last couple minutes, I'm just going to mention a website that you may not have heard of before. Human Protein Atlas is from a foundation, and it's Swedish-based, has very good pathology support. They've evaluated over 24,000 antibodies and 16 different thousand gene products. So if you have, the chances are that your gene product of interest will probably have had full staining on tissue microarrays of about 20 different cancer types. And so here's some examples of that. This is the increase in genes evaluated. This is about 16,000 over here, and antibodies tested. And here's sort of the interfaces, what you'd see it if I put in, for instance, CD247 as an example of a T cell gene. Typically we use other CD3 markers than this, but this is one. You'll come up, there's a normal tissue area here, but then there's also a cancer slot here. If I clicked on that in the cancer slot, you see the actual TMAs that were stained for CD247, and then you can click on one of them or any one of them, and you can actually sort of navigate around that image of what the staining looks like in that particular type. And so as a pathologist, I can tell that these larger cells are melanoma cells, that there's blood vessels in between here, and that most of the T cells here are sort of in the vascular areas outside of the tumor, but they're actually in that tumor. I've actually quantitated Human Protein Atlas data as primary data for papers and grants. I think most journals will view that as acceptable as long as the analysis is new and you explain how and what you did. The danger is that you don't know if the antibody staining is right. As a pathologist, I wouldn't report it if I didn't think it was right. So here, the cell size is typical of what a lymphocyte would look like. The location is typical. You can look at the normal controls to actually have someone look at it that way. But if you have a hypothesis about, for instance, HIF1 alpha, when I look at it in melanoma, it's not typically in the melanoma cells, it's actually in the macrophages and other things adjacent to it. So with the help of a pathologist, you can make your own educated, pathology on the fly guesses, but then you can work up and actually get data that you can use both in grants and papers. This is much easier than trying to arrange access to all the tissue types that you might want to do if that will suit your needs. And to be honest, the staining level here generally is also outstanding, better than in most papers. And so, again, as possible examples, you want to have a pathologist on board, and these are just examples, like for instance, you can look in the benign tissue counterpart, melanocytes, because they're skin biopsies. So the very last thing is sort of what's available at Yale. This is not going to be enough, but if you look at the Yale Pathology Tissue Services website, which David Rim runs, it's an outstanding website. A lot of knowledge there if you go and talk to folks in that unit. They will probably help you also find access to tissue. And you should realize that the overwhelming thing you should be looking at is archived surgical pathology cases. A patient comes in, has a resection. Some of that tissue is used to make a diagnosis. That tissue sits in paraffin blocks for the next 10 or 20 years. With the proper permission, you can go, which usually is pretty minimal in terms of permission. You don't always have to get full consent for that typically, but you can go and actually query that tissue. And you can do almost all the things you'd want to do on formalin-fixed paraffin-embedded tissue. So there are frozen tissue banks, the skin and lung spores in particular, other individualized banks. These are a little more work to carry out and to know who has them, and we're trying to optimize that more through, I guess, Joanne Sweezy's leading that effort. So these are all the things you can do in formalin-fixed paraffin-embedded tissue. Pretty much all the things you'd like to do. You can't get live cells from FFPE. So if you want to look at a T cell response after you stimulate it, you can't do that off of formalin-fixed paraffin-embedded. So it's a functional response. It can't really be done. But you can actually do full-on microarray data, which is pretty robust now. Affymetrix has a pretty reasonable system with really small amounts of starting RNA that will rival frozen RNA-seq data. So I just wanted to thank the folks in the lab who did a lot of the work related to the Yum lines. Great collaborators. There's many collaborators at Yale. I'd be happy to try to maybe field questions. I think it's late, so I'm not sure if you want to have a general question, maybe one. Yeah, we'll do maybe one general question. But anyway, thanks for your attention, and sorry I went a little long.
Thank you. That was very instructive. We are running late, so maybe one question? Well, I'll offer one, which is there is discussion about doing metabolomic assessments in formalin-fixed paraffin-embedded tissue, which when I heard, I thought, that seems inconceivable. Do you have any experience with that?
Yeah, so there's Vanderbilt has a very, very strong mass spec group, and they do this on a regular basis where they'll have a 50 micron spot size, which is probably about 5 to 8 cell diameters. I'm not sure that it's individual cell metabolomics, but if you have specifically metabolites of interest. You can't do the broad screens that you typically would want to do with something like that, but Rosita Lazova, who had been at Yale, worked with Richard Caprioli at Vanderbilt to do that. And I think Caroline Johnson, who's a new faculty member here, also has mentioned that she has potentially the capability to do those things. And for those of you who don't know Caroline, she also is one of the leaders in unbiased sort of small metabolite analysis in colon cancer, among other places. But it can be done off of tissue. The example I'd use is in the Yummer lines that grow out, where a tumor grows out, we see a 50 micron spot size of things that exclude white blood cells. So the question is it lactate? Is there something in that area that's